The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.
Hello, welcome to episode 10 of the eLife podcast, which is produced by The Naked Scientists. I'm Chris Smith. In the programme this month, an anti-aphrodisiac that puts males off mating with females. How a squid and a bacterium communicate, and how gaining the power of flight has affected insects' senses of smells. But first, to an experiment into evolution. Michael Cox is at the University of Wisconsin-Madison.
We wanted to understand what makes a cell resistant to ionizing radiation. So ionizing radiation is the kind of radiation that you get from X-rays. It's the kind of radiation that causes enormous damage to cells. It's important for a few reasons. There are bacterial species and also some fungal species, frankly, that are very highly resistant to ionizing radiation. To give you an idea, ionizing radiation is measured in units called gray. A human being would be killed by having a dose of 3 to 5 gray. A bacterial species that we work with, called Deinococcus radiodurans, can survive 5,000 gray with no lethality. So it's literally thousands of times more resistant than a human being would be. A second issue is just understanding why ionizing radiation causes cells to become sick. Is it damage to the DNA? Is it damage to proteins in the cell? What is it about ionizing radiation that is most dangerous to a cell and causes radiation sickness?
How have you pursued this?
Well, we realized that we could let the cells tell us what was important, basically. And you do that by doing an evolution experiment. So basically we took E. coli cells, which are not particularly radiation resistant, and we subjected them to high doses of ionizing radiation, enough to kill 99.9% of the cells. And then we grew up to survivors. And then we did it again. We went through 20 cycles of this selection. And by the time we were finished, we had a culture of E. coli cells that was almost as resistant to ionizing radiation as was Deinococcus. We did that experiment 4 times and got the same result all 4 times. And it was those populations of very resistant cells that we used to try to figure out what was important in making a cell resistant to ionizing radiation. And the advantage of doing this experiment is you don't have to look through all the genes in the cell. All you have to do is figure out which genes had changes in them.
So did you literally take the very resistant coliforms that came out of the evolution experiment and just like for like compare the genomes?
We took about 30 isolates from the various populations and obtained a complete genomic sequence of each isolate. And that told us where all the mutations were in each one of these isolates. Now, a typical isolate has about 70 mutations, and many of those mutations are irrelevant. They're just neutral mutations that kind of appeared during the selection and don't really contribute to the phenotype. So the real job was to figure out which of the mutations were really important. And by doing this with 4 different populations, we could compare isolates from different populations and find patterns. And so we focused on genes that were targets of mutations over and over again in the different populations. And we were able to home in on mutations that had a particular importance to this new capacity of the cells to resist ionizing radiation.
When you looked at the resistant forms, what changes had they adopted?
In the population that we looked at, we were able to explain virtually the entire phenotype on the basis of 3 mutations. And these mutations were all in genes that encode proteins that are involved in DNA repair. In the case of this one population, the major change that occurred in the population was to improve its capacity to repair its DNA and explain virtually the entire phenotype. Now, in other populations, there are mutations that we suspect are having a different kind of effect, making it more difficult for the proteins to become oxidatively damaged by the radiation. So in these other populations, some of the adaptations that are occurring have to do with protecting the cell from ionizing radiation rather than repairing the DNA. And so this is an example of another way to make oneself more resistant to ionizing radiation.
What do you think the implications of this are?
It's likely to give us a better understanding of what makes a cell particularly sick, what makes it susceptible to radiation poisoning. And I think damage to its proteins and damage to its DNA is clearly the major thing that happens to cells. Another implication is we should be able to generate eventually radiation-resistant bacteria that could potentially be used to do bioremediation in radiation waste dumps. I think there's potential to use some of this technology in useful ways.
Michael Cox. Pheromones are chemicals that can alter behaviour, like mating. And now researchers have discovered the pheromone equivalent of an anti-aphrodisiac. National University of Singapore scientist Joanne Yu found that male fruit flies impregnate a female fly with a cocktail of fats, which puts other males off wanting to mate with her.
Pheromones are chemicals that animals use to communicate with each other, and we wanted to see what kinds of different chemicals are used by different species of fruit flies. So the reason we thought we might be able to see something that people hadn't seen before is because of a method we worked out a couple of years ago where we use a laser to scan the surface of the insects, and by doing so we're able to detect different kinds of molecules than the kind of classical methods that are used to identify these molecules.
So that laser technique would enable you to study not just what the insect itself is producing, but potentially what another insect it's been knocking around with may have left on it.
Yes, that's right. Yes, so by using a really small laser compared to the size of the insect, we get really good spatial specificity as to where these different molecules are expressed on the surface and also where they get transferred following some kind of behavioral interaction, like mating.
And that particular laser technique doesn't just tell you where on the insect, it will actually give you the chemical identity of the substance, will it?
Yes, well, it gives you a good guess. So based on that guess, you know what is the elemental composition, what kinds of different atoms are there, and probably number of double bonds there.
When you started doing this with these fruit flies, what did you see that stood out as clearly different or exceptional compared to what previously people had managed to spot using traditional techniques to study pheromones?
So we had an expectation of the kinds of molecules we would see based on their mass. So we had a laundry list of things to look for. But what we saw that was novel was that in males only and not females, there are a series of molecules that looked a lot heavier than what had been reported before. And then when we looked at things like the elemental composition and number of double bonds, it seemed to fit characteristics of what we would expect from a particular kind of fat.
So these male flies are depositing onto the females, into the females' fats, which appear to be linked to mating or influencing mating behaviour.
That's right, yes.
So what are these fats and how do they change the behaviour of the female, and when does the male put them on the female?
They are transferring a particular kind of fat called a triglyceride. This is a common kind of fat molecule, and the males express it in a region close to their genitals. And so when they mate with the female, they're transferring sperm as well as these particular fats.
And the triglycerides go on to the female, but do they affect the behaviour of the female, or do they affect the behaviour of other males, or both?
We looked at both aspects of that. Females, after they mate, they exhibit a series of rejection behaviors where they really are not interested in mating anymore for some species. And so if a male tries to mate with them, she will kick them in the head and run away. And so we tried to see if we perfumed the female purely with these triglycerides and not have her mate with the male would cause her to do any of these behaviors, but it doesn't seem to be the case. So it seems that it's more to tell other males to avoid this female.
Let's just run over that. The female that you coat in this material, in these triglycerides, does not appear to be less receptive to mating, but she is actively avoided by males that could potentially mate with her when this chemical is present.
Yes, that's right.
Do you know how the males are picking it up or how they're responding to it? And if they do respond like this, why don't they get confused with their own smells and therefore just go into a non-mating state?
Yeah, that's a great question. We speculate because it's context dependent, and so they're smelling this smell on top of the female's natural scent. The whole bouquet of the female scent plus this added male fat is probably the signal that tells other males to avoid this female.
Now, you've looked just in these insects in this first instance, but do you think that this whole concept, for want of a better phrase, an anti-aphrodisiac, do you think this applies in other animals too, and dare I say, even in humans?
I think so. I know so because there's actually a large body of literature showing this phenomenon in other insects. And in humans, it's as difficult to say, especially in terms of chemical signals, but it benefits both the male and the female to have some kind of signal indicating a period where the female is not interested and where males won't be successful. So it seems reasonable that it would be a conserved mechanism.
Joanne Yu. You're listening to the eLife podcast with me, Chris Smith. In a moment, why taking to the air may have forced insects to improve their sense of smell. But before that, to a unique partnership between a small 3-centimetre long squid called Euprimnus scalopes and a bacterium called Vibrio fischeri. In return for sugars and amino acids, the bacteria inhabit specialised organs on the squid where they pump out light, providing the animal with camouflage. But how do these microbes talk with their hosts? Caitlin Brennan has found that molecules from the sheaths around the flagella that they use to propel themselves along are the critical component.
We're really interested in how bacteria are moving around and using a flagellum to swim through different media and different liquids, and how this behavior was important for interactions with hosts. And to do this, we were using the squid Vibrio mutualism, which is a model of host microbe interactions and is used to look at issues of host specificity.
So what did the squid do with the Vibrio bacteria?
The objective of the mutualism is to provide light for the squid, which use it for counterillumination, which is a camouflaging mechanism. And Vibrio fischeri is the only bacterium that's able to enter this specialized organ, grow there, and provide the light that the squid needs for this behavior.
So how did you use that system to understand what the flagella does normally in relation to its host?
So because of the specificity between one bacterium and one organism, we're able to probe very specific questions in terms of the conversation between these two organisms. In a way we're not able to in a more complex environment like the many, many microbes that are found in the human gut. And that conversation is very, very complex because there are so many players.
So do you think then there's some kind of signal coming out of the bacterium which is altering the behaviour of the squid and in order to achieve this mutualistic association?
So we've shown in the past that there are specific signals put out by the bacterium that are really actually common signals to many bacteria. So these include things like LPS, or lipopolysaccharide, and parts of peptidoglycan. And these are surface molecules that are found on many, many bacteria, not just Vibrio fischeri. But what's unique about these signals is where they're expressed by Vibrio fischeri and where the squid is being exposed to them. They're being exposed to these signal molecules at a place only Vibrio fischeri is able to access, and exploiting this relationship to sense when Vibrio fischeri is there.
And when you study this interaction, what is this telling you about that chemical conversation?
So it's telling us a lot about whether these molecules are the same molecules that are often causing really reactive host responses in a pathogenic relationship. If this was happening in your gut by the rotten bacterium, you'd be very sick and you'd have an inflammation. But here it's part of the natural progression of the interaction between Vibrio fischeri and the squid.
So how did you actually study this? What did you do in order to unpick this?
So we used 3 different strains, one bacterium that produces a flagellum and is able to swim, another that produces a flagellum but can't actually rotate this flagellum, so it can't swim, and one that doesn't produce a flagellum at all. Using these 3 different strains, we were able to parse apart what was actually due to the production of a flagellum and what was actually due to being able to swim and being able to rotate that flagellum. And what we found was that it didn't matter whether Vibrio fischeri made a flagellum. It's if it couldn't swim and if it couldn't turn this flagellum that it was unable to cause this host response. So we looked a little more closely at the flagellum of Vibrio fischeri, and it's known that it's covered in a sheath. And this particular flagellar sheath is a continuation of the rest of the outer membrane of the bacterium. And so what this is largely made of is lipopolysaccharide. And so we were wondered whether being unable to turn this prevented release of this molecule in a way that stimulated the host response by wild-type Vibrio fischeri.
What is the effective bottom line here then? So the bugs use their flagella to release or dispense LPS, and that the host is responding to that LPS, and in the case of the squid, for a beneficial outcome.
This flagellar sheath is something that's been very poorly understood. I found a review at one point during my graduate work that I believe was from 1981. It was the only review there's ever been about a flagellar sheath. And every single question they posed remains unanswered to this day. People have been assuming that the function of this sheath is to really prevent an immune response to the flagellin subunit. We just haven't had the tools to probe that in any bacteria that has a flagellar sheath. And these include several important human pathogens like Helicobacter pylori, Vibrio cholerae. What we were able to do was use the squid Vibrio symbiosis to highlight something that's potentially common to all these bacteria with sheath flagella and may be important in how those bacteria are interacting with their respective hosts as well.
Caitlin Brennan, she's now at Harvard School of Public Health. Finally, how insects came by their sense of smell. They actually have 2 different smell systems. An ancient one that they share with their crustacean cousins, like crabs, and a newer one that they've evolved separately. Christine Misbach.
Insects mainly use 2 receptive families. One, the family of so-called insect olfactory receptors. These are specific to the insects. The closest relatives of the insects, the crabs, do not have this kind of receptors. There is the hypothesis that insect olfactory receptors have evolved when the insect had to transition from water to land and detect airborne chemicals.
So how did you try to understand whether that was the case, that as soon as the insects left the water and came onto land, they responded by producing a new class of olfactory receptors?
Most studies on the insect olfactory receptors were done in flying insects. So nobody has ever looked into taxa that are older, for example, the jumping bristletales or the firebrats. And by comparing the results we got from those 2 higher insects or the closely related crustaceans, we hope to get new insights into the evolution of these insect olfactory genes.
So how did you study those insect species which are these more primitive, non-flying insects, to find out how they respond to smells?
First of all, of course, we had to show that these insects have a functional olfactory system using electrophysiology. The insects have very long antennae that are covered by small cuticular structures called sensilla, and the neurons are sitting inside these sensilla, and we can contact these sensilla and record from the neurons inside the sensilla.
So you were making electrical recordings from the olfactory apparatus of these insects, and presenting them with different chemicals to see if they all responded and if they responded in the same way to those chemicals across the different classes of insects you studied?
Yeah. First of all, we can use this technique to show that they can smell and also try to categorize the different neurons in the sensilla and count also the number of types of neurons that are present.
And that will presumably give you a handle on that evolutionary timeline, because presenting these chemicals to the different groups of insects, I presume what you're able to do is to show that with evolutionary time, the smelling ability becomes more diverse.
Yeah. First of all, the numbers of neurons we identified for this primitive insect were much smaller and also rather broadly tuned.
So did you then, having identified the responses to the chemicals, go to the genome and find the genes corresponding to these receptors?
We used RNA-seq, looking which genes are active in the olfactory tissue, and then searched into this data for the receptors.
So putting all of the findings together, what do you conclude?
We think the insect type of olfactory receptor is not an adaptation to the terrestrial conditions and the detection of volatile airborne chemicals, but evolved later during the evolution of insects. We think that there could be an adaptation to the insect fly, because during fly, also a much higher temporal resolution and higher sensitivity is necessary.
So taking to the air as well as taking the air. That was Christine Misbach from the Max Planck Institute of Chemical Ecology in Jena. That's it for this month. Previous episodes of this podcast are available at elife.elifesciences.org/podcast, and all of the papers that we've discussed are there and freely available too. My name is Chris Smith. This is a Naked Scientists production, and do please join me next time for another look inside eLife. Goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.